My name is Lisa Thomas, and I'm a trustee of the World Affairs Council. I'm delighted to welcome you tonight to participate in the World Affairs Council Business Forum that convenes business and thought leaders on conversations on topics at the nexus of technology, commerce, international policy, and society. It's this nexus that'll be critical for today's and tomorrow's businesses, their leaders, as well as their employees, as they strive to understand the geopolitical and policy issues that expand or limit market opportunities and impacts on society. Tonight's program will focus on the convergence of two disruptive technologies, big data analytics and next-generation gene sequencing, and the impact that these technologies will have on healthcare worldwide, including ethics and privacy challenges. I want to thank our host this evening, UCSF, and the Institute for Human Genetics, and our reception sponsor, McKinsey & Company, and its research arm, McKinsey Global Institute. Now I'd like to introduce our moderator for this evening, Michael Chewy, who is a partner at the McKinsey Global Institute. Michael leads research on the impact of information technologies and innovation on business, the economy, and society. Michael has led McKinsey research in areas such as big data, Web 2.0 and collaboration technologies, and the Internet of Things. Please join me in welcoming Michael Chewy, who will lead tonight's discussion. Thank you.
Thank you, Lisa. Thanks to the World Affairs Council, thanks to UCSF for convening what could hardly be a more important topic for us to discuss. I'm joined by an extremely distinguished panel of experts whom I'll introduce in a moment. At the McKinsey Global Institute, we've been studying big data, open data, the idea of data's impact on the economy and society for several years now. Our point of view is that the effective use of data is becoming a basis of competition across the entire economy, and it has tremendous implications in terms both positive as well as potential risks for society. Both individuals, patients you might call them, countries as well as industry sectors. And then of course, as we looked at disruptive technologies, clearly what we pun intended called next generation genomics has tremendous potential as well. In healthcare, but beyond. And so the combination of those two and that intersection is something that we want to explore today with a tremendous panel here. But just to give you a sense of the potential, when we looked at the impact, the potential impact of data, open data, some of which is genomic data, on the US healthcare system alone, not by itself, but with a number of other levers, we could see a potential impact on an economic basis of over $300 billion annually. 300 billions with a B, right? So it's a lot of potential there. So why don't I introduce our distinguished panel, whom we'll ask to have a discussion, make it informative and then entertaining, hopefully. To my immediate right, Francis deSouza is president of Illumina. It's a next generation sequencing company whose vision is to advance human health through the unlocking of the genome. Prior to Illumina, Francis was at Symantec Corporation, where you served in various roles since 2006. Dr. Tim Behrens is Senior Director of Human Genetics at Genentech. Tim joined Genentech in 2006, following over 20 years in academia, with a focus on studying and understanding the genetics of human autoimmune diseases. Recently, you've been particularly, let's hope this is true, right? Particularly intrigued by the problem of applying genetic and proteomic technologies into clinical practice. In addition to your work at Genentech, you're also an adjunct professor at the University of Minnesota Medical School, where you previously headed the Autoimmunity Center. We got that all right, right? Yeah. Terrific. And then Dr. Robert Nussbaum is a board-certified internist and a medical geneticist. I don't know what other type of certification there is, but we're glad that the board agreed to certify you. Specializing in the care of adults with hereditary disorders. Also chief of genomic medicine here at the UCSF Medical Center and co-director of the program in cardiovascular genetics at the UCSF Heart and Vascular Center. In addition, you're a member of the UCSF Institute for Human Genetics, also host for us here, where you're studying how genetic and genomic information can be used to improve health, and also a member of the Institute of Medicine, and co-discoverer of the first inherited form of Parkinson's disease. And then prior to joining UCSF, Chief of Genetic Disease Research Branch of the National Human Genome Research Institute, one of the National Institutes of Health. And there are indeed different institutes for those of us who don't pay that close attention. All right. So with that, obviously a terribly qualified panel, but let's start with just a very basic topic of discussion. So for each of you, coming from diverse and yet related backgrounds, what do you mean when you hear, or what do you think of when you hear the term big data or big data? And how does that intersect with genomics? What are the things that you're seeing? How do those things put together actually create benefit? So Francis, let's start with you.
Sure, and I think what's really exciting about this topic at this time is I feel like we're really seeing sort of the intersection of two trends, two industries that are going through their own inflection points. So, when we think about big data, there are several things that make up, big data. One is the sheer volume of information we're talking about, right? So whether it's genomic data or sensor data from the Internet of Things, the amount of data that companies have to deal with today doubles almost every year, every 18 months. And we're seeing that play out now for several years and continue to play out. So it's the amount of information. Secondly, it's the volatility of the information. So the information that you have to deal with is changing very, very quickly. So it's not static information that you can just store. And then third, very often the data is unstructured. So it's not the data that sits in a database with a very specific format. And so those things are coming together. We're seeing more information created than ever before. We're seeing it change more frequently than ever before. And it's unstructured. So that's happening with big data. Genomics is going through its own sort of inflection point where we're now seeing the era, I believe, of applied genomics, where in 2003, we sequenced the first human genome. That was a huge achievement for humanity and for science. And there's been a lot of work since, and now we're starting to see genomics enter a number of applications, whether it's medical applications like oncology or reproductive and genetic health or forensics. And so that's a number of technology trends that are driving that, and it's intersecting with big data, and the amount of things that we can now do or start to think about doing is fantastic.
Can you say about what are some of those things that you can think about doing?
Sure. We are now looking at, and I'm sure we'll spend a lot of time talking about oncology and the treatment of cancer, but cancer is fundamentally an acquired genetic disease, right? And so now there are a number of ways that we're looking at leveraging genomics to really revolutionize how we screen patients for cancer and seeing if we can look at a patient's blood to identify very early on if there are traces of cancer. We're looking at very differently how we can prescribe a therapeutic regimen. So based on the genetic profile of the tumor and the patient, start to understand what combination of drugs would be most effective against that tumor for this patient. We're also thinking about how we can change the way we screen for recurrence of that cancer. And again, looking potentially in the blood for that sort of genetic profile of that tumor. And so that's an area that we think will be very radically transformed over the next few years with genomics entering that. But it's not just medical applications. I mean, we're looking at forensics, for example, and being able to identify from a sample of blood, for example, of other body fluids, be able to go from that sample to a facial profile of the people whose genetic material was in that sample very quickly. So it's cryptography, it's forensics, it's agriculture. There are a number of industries that have now got access to genomics because we're hitting price points where it's attainable for that industry, and that's very exciting.
How much does it cost now?
Well, in January, I announced that you can do a whole human genome for $1,000. Now, when you consider 11 years ago to sequence the first human genome, it cost $3 billion and took 15 years. So it's less than 1/millionth of the cost it took 11 years ago.
It's quite a discount. Tim.
Yeah, so in my role as sort of overseer of human genetics at Genentech, big data sort of is equivalent to genomic data. And thanks to Illumina and companies like Illumina that have really driven the cost for generating this data to an inflection point, where it really makes a lot of sense now that every person that we enter into a clinical trial, and there are over 200,000 people in clinical trials just for the Roche Genentech group alone, and multiply that times many, many pharma companies. Essentially, the price point is there to have this as just part of an ongoing clinical trial. And when I think about big data, it is both the big data that lies outside in various academic centers, in various repositories of this kind of data. There's some real issues with accessing that data, with even moving the raw data. It's just such a tremendous amount of data that we have to come to grips with. How do we actually access, move, and analyze that kind of data? But then there's also the data that's being generated internally, and I can tell you that our internal just hardware is really stressed and growing rapidly just to accommodate all this genomic data primarily because of the storage costs and the storage issues around the raw data. So, but ultimately it's translating all this genomic data into something that's useful. And for us, the two most useful aspects of these kinds of data are, number one, when we take new medicines into the clinic, really trying to understand who's going to respond and who's not going to respond to a drug. And that goes for cancer medicines, that goes for medicines into various other diseases. And we have more and more evidence that the genetic data, the genetic code is telling us a lot that helps us understand who is the right responder, who's the right patient to treat with with a particular drug versus another drug that might be better for that patient. And there's a whole lot of effort going on, I think, throughout pharma, really trying to take advantage of this genomic data in predicting treatment responses. And then the second major area is really to use this data to better understand just human disease and what's the drivers of human disease. Not just cancer, which we know has a very strong genetic cause, but diseases like asthma, diseases like rheumatoid arthritis, diseases like inflammatory bowel disease, psychiatric disease. We know there's a genetic component to these diseases. We think that this genomic data is going to tell us really important things about the drivers of these diseases. And if we can understand that better, we can then find good targets for therapies, for the new therapies that'll hit patients in 10, 15 years.
So it's interesting because as you described it, there's a lot of data associated with just the genomic data, but if you're actually going to figure out what's going on clinically, you need a whole bunch of other data as well to add to it. What's the other data that you need?
So, as part of a clinical trial, you're generating very, very extensive what we call case report form data. It's the clinical data around that patient, how long have they had disease, what kind of organ systems are involved, what kind of drugs have they been on, the basic laboratory test, the common laboratory test that your doctor would order with a yearly physical, possibly some special tests, imaging tests. In a way, that data is very complex, but it pales in comparison to some of the genomic data, at least in terms of just quantity of data. So we end up with very high dimensional data sets where we have one patient with a tremendous amount of genomic data, a fairly good amount of clinical data, some laboratory data, and then you can then add other things to that, proteomic data. So what do the proteins in the body look like? What does the microbiome look like? I think there's a lot of interest in exploring that going into the future.
Say what that means. What's the microbiome?
So the microbiome, it turns out that we have microbes living in essentially every surface of our body and most cavities of our body. And some people have estimated that there's more genomes of microbes in our body than there are cells, our own human cells. And so we're just coming to grips with some of the complexity there. And these next generation sequencing methods, such as pioneered by Illumina, are really just opening up new fields there that, again, are creating very interesting data sets, but they're extremely large, they're very complex, and some at some point we need to integrate all this data.
So for the geeks out there, how human genome takes about how much?
Well, you can, if the raw data, all the BAM files, I think for one genome, it's about 1 terabyte. It might be a little bit less, but that's when we move this data around, we can't send it through the internet. So we have to send physical hard drives. So if we get individuals sequenced either from our clinical trials or other research subjects, and we have those sequenced at Broad in Boston, we have FedEx sending us large crates filled with hard drives to move all this data around. So that's one of the practical issues of dealing with these large data sets.
Bob, what are you seeing from your perspective?
So for me, it's indeed it's true that we can generate large amounts of genomic data, but our ability to actually read it, interpret it, is very, very primitive. The part of the genome that actually codes for genes, codes for proteins, which is about maybe 1 to 2%, is just a small fraction of what's there. And you can look at an entire genome, and our ability to extract useful information from it is at an extremely primitive stage. The only way that we're really going to be able to understand, to be able to read and interpret a genome, is to be able to link genetic information to what's going on in the person whose genome that is. And that kind of data I think of as being really big data, because it involves having a very clear picture of that person's appearance, experiences, environment, what they're exposed to, a whole variety of things, perhaps even going back to what they were exposed to when they first went through the birth canal. So there are really critical amounts of data that we need that are unstructured, they're complicated, they're not well structured at all. A lot of it is buried, is either unavailable or is buried in text files in doctors' records. Extremely difficult data to acquire. But only if we can get that data and link it to the genomic information are we really going to be able to understand what the genomes are telling us. I mean, I tend to think of the genome, people have used lots of analogies, and analogies are dangerous. But I think it was during the time of the Genome Project, it was described as the Holy Grail. It's not the Holy Grail. As a blueprint, it's nowhere near as definitive as a blueprint. I think of it more like a high school football play that the coach puts into operation. But the field could be wet or dry. The quarterback could be either in a good mood or a bad mood or hungover. There's all kinds of things that can, and of course that doesn't take into account at all what's going on with the opponent, with the other team that you're playing against. So it's sort of a general description of the way things perhaps could go, but being interacted with and being changed constantly by the environment and by a whole variety of other things that are going on. And we cannot really interpret genomes very well. We need this other data, and there are powerful cultural, structural, ethical, and regulatory areas that control our ability to access exactly the data we need to make sense. I mean, Tim makes a great point about being able to connect data from clinical trials. But the vast majority of data in this country are not people in clinical trials. They're people that are going to see their physicians. And this information is buried, some of it on paper, some of it in electronic medical records that are, I'd say, primitive at best. They're more like electronic paper. And so making that work is a huge challenge. And to me, that's what big data really means, getting at that stuff.
So what's the current state of the art, and what's the potential going forward? And then let's talk a little bit about some of those obstacles that you mentioned there, or constraints.
Well, the current state of the art is that I think just in a few years, because of the pressure coming from the federal government, essentially, and both pressure and carrots and sticks, both, there's been a huge shift towards the adoption of electronic medical records. And this has just happened really just in the past few years. Despite that, most of the data in electronic medical records is still not in a structured format that's easy to get at. And so there's a lot of effort that's being made at text mining and at being able to extract useful information from text. But let's go through what are some of the issues. I mean, first of all, patients don't go to see their doctor because they are implicitly signing up for research efforts. They're going to see their doctors because they have an issue and that they would like to have help with. And so there are concerns, there are fears. We have, for example, the Genetic Information Non-Discrimination Act in this country, which says that a genetic change is not a disease. And you therefore cannot be discriminated against for what's in your genome in employment and in health insurance. Nothing about life insurance, nothing about long-term care insurance, nothing about disability insurance, and people are concerned that they're going to be at risk for that. The whole idea of being able to extract this information from medical records requires a time-intensive and extremely expensive consent process. There are some places, Vanderbilt University, for example, which has instituted a system called opt-out, where people who go to seek medical care at Vanderbilt can check if they do not want their information used. And 5% of people check that box. 95% say, fine, it's okay. But questions are raised as to, is that really informed consent? Is that similar to what we do when we download a software package and you're supposed to say you agree at the bottom? Who reads that? I don't. Do you? Do you read that? And so the question really is that really informed consent? There are other issues. For example, there are some private laboratories who feel that their testing information that they have generated, for example, suppose they had a monopoly on testing certain genes, that information is proprietary. They're monetizing it. It's intellectual property. They do not want to share it. I personally think that that's an ethically questionable position to take, but that's my opinion. Other people have other opinions. So there are lots of issues involved in trying to get exactly that kind of data. If you've ever worked in a clinical laboratory, you may get a sample on a patient. It says, we would like to rule out hereditary breast and ovarian cancer gene mutation in BRCA1 and 2. That's the sum total of the information that clinical laboratory gets about that patient. Rule this out. You actually know nothing about that patient, really. Yet that's what the laboratory has. So who is going to provide the information about that patient to a database that allows us to link a variant found, let's say, in BRCA1 and the clinical picture? Is this a patient who developed bilateral breast cancer at age 32? Or is this a patient, what's going on with this patient? Who gives the consent for that? Where does that information reside? Who has the time and the energy to enter that information? And how does the patient participate in making that decision that they want their information used? These are all, I think, big questions that need to be addressed, and they have not been.
Let's keep exploring this, but before we do, I'm going to let people know there will be an opportunity to ask questions. I can see questions already formulating in people's minds. Those who want to tweet the questions, you can tweet at world_affairs and please hashtag big data. Tim, we talked earlier about, Bob mentioned this too. When are people going to get that information? If they're sequenced, or back to them, and what implications does that have? I'd love to hear what you think about that.
I think that's also a conversation that is going on right now, more generally. So if somebody goes to a company like 23andMe and requests them to take a blood sample or a saliva sample, generate genetic data, they get that data back, sent to them by email, and they have their genetic marker data, about 1 million genetic markers across their genome. And that's a clear case where they pay a purchase price, they get the genetic data back. Right now, they're not getting a lot of information about what any of that means. Essentially, it's just raw data at this point because of a FDA hold. But I think the broader question is when patients are seeing their doctor at an academic center that's interested in doing this kind of work, they sign a consent form. Generally speaking, most of the consents say that we're not going to give this primary data back to you. You probably will not benefit personally from, the research that's done on your sample. And even though that's the sort of written out in the consent, most patients are more than happy to, participate in research because they have a particular disease, or they have a family member with a particular disease, and they're very motivated to provide and really do anything they can to advance research. What will happen now that we can more routinely generate this kind of very deep data, and how valuable is that? Right now the value is being sort of estimated for an individual patient, that individual level data, and for some diseases where there's a very strong link between the genetics and future risk for disease, some of that data is actually actionable. So I think we're in a transition period where in the past, this kind of signing up for a research project, not really caring if the data came back to you, I think is going to get phased out, because I think patients are going to want to be more involved and have access to some of that data. On the other hand, and Bob can probably talk to this even better, there are certain patients who may choose not to get genetic information particularly back from the doctor. They might not even want to know their blood sugar level, and the discretion has always been that a patient sort of makes that call as to whether they want to receive information or not. And I think there has been some recent discussion at very high levels in the medical genetics community about this issue. You may want to speak to that.
Well, yeah, the issue is really around, suppose you go to the doctor and you want a specific genetic question answered. Suppose you have a child who has a developmental delay, and what you would like to know is whether there's a genetic basis for this child's intellectual disability. And in doing that test, you discover that the child happens to carry a mutation that is known to cause early onset colon cancer. That's not why you brought your child to be tested. In fact, this early onset colon cancer doesn't occur in childhood. It occurs in adulthood. Yet, here you are with a result which could be of great significance to the child later in life, and could be of significance to one of the child's parents, from whom the child may have inherited this change. Yet that is not why the child was brought to be examined. And so the question is, what is the consent that a person gives when getting such a genetic test? And are there certain things you find, even if they're not what you're specifically looking for, that are so important and that you as a physician feel are so actionable and so important for this family to know, that you would actually tell the family, even though it's not what they originally had wanted to know? And should we be asking them up front, if we find things like that, do you want to know? And should they be consented up front for this list of 4 dozen, 5 dozen different things which some group of doctors somewhere, myself included, thought should be important and people should know about? This is a huge debate that's going on right now, the so-called incidental or secondary findings area, because once you start doing whole genomes, you find stuff.
For instance, you're in the business of creating machines that do whole genomes. When are people just going to get this as part of their physical, for instance?
I think we believe sort of 2 things will play out. One is that what you will see is different industries will adopt sequencing at different rates. So I'll give you an example. Today, when you have a couple that's pregnant, it used to be that if you were in a high-risk pregnancy, so if you were over a certain age for the woman, or you had certain family factors, in some cases you'd go and do an amnio. Now, for those of you that know that procedure, it's not a painless procedure, and it carries some risk to the baby when you do the procedure. And what we found actually is that you can do a non-invasive prenatal test, taking blood from the mother, and actually get a more accurate result by sequencing that blood. And what we've seen is, that has now become, I think, one of the fastest adopted diagnostic tests in US history. So it's played out over the last 18 months. And now, certainly in California, if you go in and you are in that high-risk category, it's rare that you will go and do an amnio. More likely, you will do what's called NIPT, non-invasive prenatal testing. And so in that segment, we've seen very, very rapid adoption. And so we'll see, I think, different segments sort of play out at different speeds. And that's true in medical areas, but it's true, again, in things like forensics and sort of non-medical areas, well, agriculture and so on. So I think that's sort of one dynamic that we'll see play out. The second is, at this price point, we're seeing a lot of research that's enabled. So not necessarily an applied market, but we're seeing, for example, in England, there's this project, GEL, which is Genomics England project, where the country is actually kicked off a very large-scale project where they want to sequence 100,000 genomes and try and do discovery. And so you'll see these kind of population sequencing efforts kicked off at a national level. And frankly, those are important because that type of research is essential to discover what we can find in the genome. What is the utility of all this information that we're getting? And so we'll see those efforts kick off as well.
So who owns this genetic information that gets generated out of one of these projects?
It's a huge question. I think today, for example, if you go into your doctor and you go into a cancer center and you get tested there, well, they have the information in much the same way today that they have your health record. And so, I think that's sort of one way it's playing out. But there is an open question around, well, how much of that ownership should go to the patient? And I think that's playing out not just for genomic data, but in general with healthcare data about how much does the patient own beyond just giving consent, right? How much can they actually take away with them? How much do they have access to it? Where it is a population effort funded by a government, and in some particular cases, the government is getting the right level of consent from participants in that study, and in some cases making that data available in an anonymized way to researchers within that country, for example. And so the ownership models, frankly, are all over the board right now. And if you are a consumer and you go directly to a company like 23andMe, you get the data, but the direct-to-consumer company may actually also own the data.
Mm. You actually have to read those.
You actually have to read those policies,
I actually should make a comment. I mean, for example, the Genome England, the reason that project is as interesting and important as it is not just that they're sequencing these folks, is that it's linked to their medical records, because they actually have a medical care system, which we don't have. And so, I think it's really important, and I'd just like to mention that UCSF has a very strong collaboration, the Institute for Immune Genetics and its director, Neil Risch, with Kaiser, Northern California, which has also collected well over 100,000 individuals with consent to allow access to their medical records and connect it to, at first, what we call genotyping information, and eventually will be sequencing information.
Say what genotyping is?
So genotyping is just looking at specific points in the genome where there are known variants that have in the past been associated with particular disorders or particular traits. They tend to be very common variants that have very small effects. And we're missing a lot of the genetic information. They call it the dark matter of the genome. We're missing a lot of that, and that can only be picked up by doing sequencing.
Yeah, other now junk DNA, which is not so junky.
Yeah, although, Sidney Brenner made the point that junk is okay, it's just not garbage DNA. Because the difference between garbage and junk is garbage is the stuff you throw out. Junk is the stuff you keep in your cellar or your attic, because you might need it someday.
That's good. So we've talked about who might own the data. Tim, who gets the value that comes from actually having this data?
Yeah, I think that's also another open question. The Supreme Court has recently ruled that genes are not patentable. That had been an open question until very recently, and now I think that that's settled law.
Except in Australia.
Genes, but not constructs made from genes.
Yes, exactly. But it takes it to a certain level, and I'm forgetting your question now. I just got off track.
Who gets the value from all this?
Yeah, so the value. So because the genes themselves are not patentable, I think that it really opens up a playing field for academic centers, for groups who have access to this kind of data, for commercial entities to take this data and reduce it to practice, reduce it to something useful, reduce it to a diagnostic test that actually helps patients in the clinic, where clearly a diagnostic company can benefit from that. For the academic groups that define interesting points and targets for therapy, they can work with their pharmaceutical group within the university, start putting chemical matter together, put a patent structure around that. And the academic centers can then, and there's a very active group here, licensing this intellectual property out to small biotechs, large biotechs, investments of large pharma, large biotechs into universities like UCSF to foster the kind of research that eventually yields targets and drug matter around that. And then clearly pharmaceutical companies, biotech companies that are trying to wrap their mind around this, all this data. Ultimately, that's our goal. That's why I went to a company like Genentech, was that I could really translate genetic data into medicines for patients. And we have a lot of people at a place like Genentech who are extremely passionate about that. And yes, it is a commercial entity. To keep the doors open, we have to sell drugs. It's a model that not everybody is exactly thrilled about, but it is a model that works and can drive real value and can drive innovation. And so we have a lot of unmet medical need out there. There's a lot of diseases where there just is really nothing that changes the course of disease. And the neurodegenerative diseases are a really good example. So in an audience like this, 1/4 of us carry at least one copy of a genetic factor called ApoE4, which triple or quadruples your risk for Alzheimer's disease. 10% of the population is going to get Alzheimer's disease, and we just have nothing available. So we need commercial entities, I think, that are really going to invest the money to try to take genetic, genomic, big data and translate that into targets and pathways that might actually make a difference for some of these diseases where we really don't have anything that's game-changing right now. And I would say that that's what gets me out of bed every morning, is the chance to really participate in a big team effort to try to find those kind of medicines. And ultimately, it's the patients, I think that benefit if we can really translate this data into something useful and meaningful.
Let's say I had a particularly cool gene. Could I monetize that in some way?
I don't know exactly how that would happen. Anything's possible.
All right. We're actually getting some questions from our audience here on the famous World Affairs Council blue cards. So let's ask away. How big a risk is too much information? As access to genetic information increases, should we worry about misunderstandings or misuse of that information? Francis,
I think that's a really important question. I think that we focused a lot on generating the data, sort of managing the data, extracting insight from the data. But one thing we didn't talk about is how that data is actually communicated to a consumer, a patient, and that is very important. We think that certainly for the medical applications, genetic counselors, your doctor has to be part of that communication, because otherwise it's really hard to interpret. Being told that you have a 11.3% chance of getting something when the population average is 9.6. What do you do with that, right? And what we don't want it to be is become some kind of molecular horoscope where people are trying to read the tea leaves and figure out what to do. And so I think it's not too much information, maybe, as the question asked, but it's the interpretation of that data. I think there'll be a big need for genetic counselors, for medical professionals to be in that dialogue.
And we are just naturally bad at statistics.
We are naturally bad at
So
statistics, yeah.
I think that's an educational need that's out there.
I was going to say, at this point, in addition to other sorts of things you could think about misuse for discrimination in a variety of insurance settings that are not protected, I'm particularly concerned about the overinterpretation of genetic variation, and it's a certain, almost a level of hubris that we really think we know what's going on when we find the change in a gene without having the real foundational information we need from patients. You know, sort of the difference between a change in a gene being deleterious to the function of that gene, harming the function of the gene, versus actually disease causing. And we have many examples where you can change a gene in a way that makes it quite non-functional, but in fact the person is perfectly fine. And so once again, it comes back to, I'm harping on this, if we don't have participation of the society in letting us connect their information to their genome information, we're not going to be able to read this properly and use it properly.
Is there a challenge with that right now in terms of how we've set up the constraints, either regulatory, legal, et cetera?
Well, I mean, there are important legal constraints. There are, you know, things like the HIPAA law about not, you know, people do have right to privacy, and there are very strict confidentiality rules, so that there's only two approaches to this. Either people have to participate in it themselves. If you give up your own information, you've given it up, you've done it. Or we have to have appropriate consent, that someone will come to you and explain to you in detail what it is that you're being asked for, why you're being asked for it, and how it's going to be used. That is labor intensive and expensive. So I'm a real strong believer in patient or social participation in this kind of research, and I think we've only just started to get that going. We need much, much more of it.
So you have telepathically segued into the next question.
Excellent.
What role should the public play in governing large collections of big data, including genomic information, in both the research and clinical contexts?
Well, there's a whole area now. First of all, I should mention that when the Human Genome Project was started, it was written into the law that 5% of the budget had to be dedicated to the study of the ethical, legal, and social implications of that research. I know of the NCI certainly didn't have that written into their National Cancer Institute budget. And what that has done is it has spawned, I think, a really enormous intellectual capacity in thinking about these problems. And one of the areas that ELSI researchers, this ethical, legal, and social issues, is the acronym is ELSI. One of the areas that they've gotten very interested in, and I think have done a lot of good work, is in community engagement. Reaching out to people and understanding, what do you think about all this? And under what circumstances would you like to participate?
So how do they do that?
Well, for example, for the Northern California Kaiser and UCSF collaboration, they went to Kaiser patients and had focus groups of various kinds, cutting across age, cutting across ethnic backgrounds, cutting across social economic status, and engaged them and asked them, this is what we were thinking about doing. How do you feel about this? Would you let us use your information if you were asked to consent to this? And I think that's really a critical component. It's very, very important. I think that one of the problems we have today is a disengaged citizenry. We're disengaged from a lot of things. We're disengaged from politics. We're disengaged from many aspects of what goes on in society. And I think we're disengaged with genetics and biological research, and that needs to be reversed.
You left us in suspense. What did the focus group say?
Oh, the vast majority of the time, they say, as long as it doesn't actually cause me harm personally and forwards our understanding, we're all for it. I mean, people, when asked, they tend to be overwhelmingly in favor of participating.
Tim, maybe I could ask you to take this one because you'd already started discussing value. So please discuss the issue of how big data is being monetized. In particular, the value of sharing data to advance understanding versus the value of keeping data proprietary in databases?
I think that's a very important point. And as these repositories of data get built up, some of them are in the public domain. There's repositories like dbGaP, which is a NIH-sponsored repository for genetic genomic data, sequencing data. Just a recent policy for genomic data sharing has been released by the NIH, which is very forward-thinking, I think. It's also requiring that consents be very explicit about how data is shared so that people understand that their data is going to be shared. I would say at Genentech, we believe very strongly that the genetic data that we generate should get out into the public domain. And so we have been depositing, as we publish studies around the work that we do, we deposit our data into publicly available databases. The only way that we're really going to, I think, wrap our minds around what this data is trying to tell us is by putting this data into publicly available and publicly accessible databases. Now, what does publicly accessible mean? I think there's a difference between a database where just anybody can, type in and say, I'd like to pull down all the Alzheimer's genetics data, and it could come from anywhere in the world. The way dbGaP is set up is that it's really set up for scientific researchers who have, they put a request in to access that data. Their request has to be consistent with the consent that the patients signed when they gave their sample up. And so there's a panel that actually reviews all the requests of this public data. And I think the best work and some of the biggest advances are going to come from large collections of these data, and I think those are going to be in the public domain. I think there may be groups who want to have a privatized batch of data, and I think they're going to have trouble keeping up with these more publicly accessible data sets.
Interesting. So you're saying that this type of open data, which we've studied as well, is just because of the size and scale and scope, is going to outpace the proprietary databases?
I think so, because there's just so many more so many academic centers. The NIH is putting a lot of money into generating this data. There's also a lot of emphasis on making sure that this data it's so valuable. And these data sets are not sort of what I would call dead data sets. Like you do an experiment in a mouse and you generate data and it's, or a set of mice. Yeah, the mice are dead, the data's dead, it's just not, these are living data sets. So as you start with 100 patients with Alzheimer's disease and clinical data and genetic data and genomic data, and that's of some value, but as you add the next 100 and then the next 2,000, and then that data set keeps growing, that even that initial data that was placed into the database is in some ways just as valuable as the last bit of data.
Plus, you're going to revisit the genetic information and be able to extract much more information.
Yeah, exactly.
You know more, yeah.
As the algorithms improve. So these are living data sets, and they have to be. You have to be able to analyze this data together to really extract the maximal value. And my sense is that the public data sets are going to be the most valuable.
But here's another example of where I think we have a regulatory obstacle, and that is the institution of IRBs, institutional review boards, who oversee the human experimentation. Extremely important because we have many examples of abuse in the past. On the other hand, you want a big data set. You want to collect information from 30 different sites. You have to get 30 different IRB protocols approved. Yeah. Now, University of California, somewhat forward-thinking, has made a reasonable single IRB procedure if you're at any of the UC campuses. It's not quite that streamlined, but it's close. But if you go outside UC, each IRB. That I think is an enormous fragmentation of the oversight of human subjects protection, and that's an area where I think we can do much better.
What's the solution?
I think the solution should be to have an IRB that is accepted, so that you say, here's the primary site that's organizing this, they have overseen this, they approve it, you should approve it too. You don't have to wordsmith, that the there really shouldn't be a the, it should be an a. That's not needed in order to protect human subjects.
When I was once in academia, I remember they had margin requirements, which was terrific.
Well, and just, we face this in the global context. So this is a very US-centric
Yes.
sort of view, but in clinical trials for pharma and biotech are going all over the world, and each country has their own very specific set of rules and regulations. They're almost like independent IRBs. Some countries will not allow any genetic testing to be done at all, period, banned. Other countries, you can do genetic testing, but the data and the DNA sample can't leave the boundaries of that country. So it's just,
Most countries, in fact, are like that.
Yeah.
And then that's also, and the flip side is true, that there are countries where, because of a single payer system, for example, where they have a chance to really move quickly ahead compared to the United States.
Francis, why don't you take this question, which is an argument against testing. Could we potentially overburden a health system, where, arguably we're already spending more in this country as a percent of GDP than any other?
Sure. I think one of the big opportunities in using sequencing is actually to limit the overuse of medication and overprescription of certain therapeutic paths, right? So, for example, if you know a person isn't going to react to a particular drug regimen, then don't prescribe it. And there's been some recent work that's come out even over the last week that shows, certainly for cancer, for example, there is sort of massive, overprescription of drugs that aren't effective. I'll give you another example in IVFs. So if a couple is trying to get pregnant, and today, about a quarter of all IVFs result in a baby at the end of the process, right? And so one of the things you could do, for example, is you could actually do sequencing of the fertilized embryos. And then what we know is that if there are chromosomal abnormalities in the embryos, they're vastly more likely to result in a spontaneous abortion. And so the carrying mother will miscarry. And so what doctors do is they plant multiple embryos with the hope that, well, they'll get one baby, and very often you get multiple births. But if you could look for that up front and say, look, these are most likely to result in spontaneous abortion, so let's pick the embryos that aren't, you could actually burden the healthcare system a lot less. Another example is babies that stay in the NICU for more than 7 days, the so-called undiagnosed babies. And what happens is those babies very often go on a multi-year odyssey of trying to discover what is the issue with the baby if it's failing to thrive. And what we find now is that, and that's a huge pain for the child, for a huge burden for the family, and a vastly expensive process and journey. But what if you could sequence up front and sort of avoid that odyssey and be able to say, this is what we've got. And we're finding that in some cases, some institutions are getting success rates, north of 20% and be able to 20-30% in terms of being able to diagnose up front and avoid that multi-year odyssey. And so there are many specific examples where you can say, look, we can be a lot smarter and a lot more effective and drive much better outcomes at a lower cost by using this sequencing data.
Yeah. Very good. Here's a relatively specific question. So maybe I'll ask Francis, but others feel free. What do you think of the role of BGI, which is Beijing Genomics, is playing in the genomic sequencing and research? Is it a collaboration? Is it a competitor? So maybe someone can explain what that company is and why some people think it's important. Yeah.
Sure, I'll jump in. So BGI is a company that's based in China, and what they do is they offer sequencing services. And so, for example, they're a large customer of Illumina and our sequencing machines. But they also bought a company here in San Francisco called Complete Genomics that does its own sequencing. And so the intent is they'll have, likely their own sequencing machines. we think it is it's certainly a player in the global sequencing services market. It has been an aggressive sort of expander into that market. And so I think that's our view of it.
I would say we're glad that BGI is around, because BGI is a competitor, and BGI, because they're a worthy competitor, is going to hopefully keep prices down. and so if Illumina had no competition, and they have plenty of competition, by the way, but we love BGI because BGI is going to be fighting for cost reductions, and we can all benefit. All the customers can benefit from a good, healthy competition.
Yeah, it's a vibrant, thriving, competitive ecosystem.
Here's a question that's relatively personal, so let me make it a little bit general. But do you have any personal stories about genetic testing, and what you or someone else was able to learn, and how that might have affected your life?
So personal meaning my own individual testing?
That's how it was originally asked, but I'm going to open it up to, if you want to broaden that to another story.
I'm 100% Ashkenazi Jewish, and I had an aunt with ovarian cancer, and I had a grandmother with pancreatic cancer, and I was concerned about BRCA1 and 2 mutations, and so I had myself tested. And I don't carry mutation in either one of those genes. I don't carry any of the 3 Ashkenazi Jewish mutations that are common in BRCA1 and 2. And I remember a certain sense of, I don't know quite where to put it. Accomplishment is not the right word. It's more a sense of responsibility, that I wrote a letter to my children saying, I was tested for these 3 genes, for these mutations. This is why I was tested. I don't have them. And since you are my children, and that I'm quite sure of, I'm passing this letter on to you. So that's a personal story.
What would the implications of having those, one of those genes, what would that have meant?
It would have meant that as a female, you'd have a 60 to 80% lifetime risk for developing breast cancer, and at an earlier onset than the 12% background risk, a 30% or so risk for ovarian cancer, which is 15 times the general population, also at an earlier time. Also an increased risk for prostate, pancreatic cancer, melanoma. And they're very serious. They're serious enough that the country of Israel is discussing sequencing all people in the country, the entire country, for whether they carry one of these 3 alterations in the BRCA1 and 2 gene, because it's present in a 3 to 5% of Ashkenazi Jews, has a very high penetrance. And the big debate in Israel now is whether they should be sequencing men and women, or just women alone. I think that's just an amazing discussion, because it completely ignores the genetics, which is men pass half of their genes on. So it's it's a fascinating discussion.
Tim, Francis, any stories you want to share?
Well, I did sign up for one of the personal genetics companies to have the service to get back my million SNPs. And when I looked through the diseases that I was most susceptible to, it was very informative for me. So it turns out that I did work on autoimmunity genetics, still have a very strong interest in autoimmunity, because virtually every other person in my family has an autoimmune disease. Rheumatoid arthritis, type 1 diabetes, alopecia areata, it's just filled with autoimmune diseases of all stripes and types. And the strongest risk, based on my personal genetic profile, was for a variety of different autoimmune diseases. So I was pretty sure they hadn't switched up my sample. And then one of the sort of interesting parts about it, another part of the report was the mitochondrial DNA. So mitochondrial DNA is passed from your mother to, it's always maternally transmitted. And my mother has her mother was from Finland. And she actually carries a keychain. So her mother's from Finland, her father's from Germany. She carries a keychain that says 100% Finnish because so she denies her German side. And so when I got my mitochondrial haplotype back, it was a Finnish mitochondrial haplotype, very strongly enriched in Finland. And so, I'm connected back to Finland, and it all my family history, made a lot of sense to me. So in some ways it was, confirming things that I already knew, but it was an interesting odyssey nevertheless.
Yeah. Having discovered this propensity for autoimmune, what did you do with that? I mean,
well, it just sort of answered some questions relating to our family, and but also, just in the spirit of the work, the research that I'm continuing to do at Genentech and developing drugs for autoimmunity, I think that there will be a day where we will be able to understand enough about the genetics and the environmental factors that we will be able to identify at-risk people for a disease like rheumatoid arthritis or lupus or one of the other autoimmune diseases, particularly the more severe types, and be able to pick up, say this is a susceptible person, let's start looking for signs and symptoms of disease really early on in this person's life. And we know that many of these autoimmune diseases, you have autoantibodies, little biomarkers in the blood that occur years before you ever have a clinical sign or symptom of the disease. And some of them are very specific for, if you get this, you are going to get the disease in a few years, and or a very high chance. It's never deterministic, to your point.
Yeah.
But I think if you have the right genetic factor, and then you start seeing biomarkers and pathways in the blood that are saying this person really is at a very high risk, I think we can get to the point, not too far down the line, of saying we're going to treat that patient before they actually ever get disease. And I'll bet that it's going to be a lot easier to treat and put into remission sort of preclinical disease than once, a disease is, especially these autoimmune inflammatory diseases, once you start getting destruction of tissues and all the secondary sequelae, they can be very difficult to treat. And I think the hope is that a lot of this genomics work, integrating all this big data will actually, put us in a place where we can think about preventing disease.
terrific.
Yeah.
Francis, a story? Otherwise, we'll go straight to the lightning round.
Just sort of a couple of quick ones. One is, and unfortunately, I think it's a story that many people sort of experienced, which is a close family friend, a woman in her early 30s, got small cell ovarian cancer, sort of a very aggressive form of ovarian cancer, and it was just a horrific sort of tragedy. It really does sort of put the urgency in, the work that I think the entire industry is doing to, screen for that disease earlier, to, really identify what course of therapies would work. And, the disease is awful, but what the patient goes through in a lot of cases with no idea whether it's going to work or not, or it's not, or if it's working. And so that's sort of my sort of personal intersection with, look, this is important. We need to do this. The other one, on a lighter note, is my mother's Ethiopian, and so I've known this my whole life, and so I did the 23andMe thing, and I noticed that there's a big sort of presence of Yemen, a specific tribe in Yemen, I guess, is a very distinctive sort of genetic group that I carry, and I was talking to my mom about it, and she's like, "Oh, that's why they call my mom the Yemeni." Maybe why, Mom?
Let's finish up with just a quick round of questions here for each of you. What worries you most about big data and genomics? Francis?
How fast we can get to use it and get to utility.
Analyzing this data and making sense of it.
Getting the patient's information hooked up and connected to it in a way that we can really start to read and understand this information.
What's the next disease that will most likely be cured through big data and genomics?
Good grief.
Cured.
Cured, huh? I'm going to go out on a limb. I'm going to say type 1 diabetes.
It'll be one of the cancers.
I think it'll be one of the cancers too.
What company other than your own would you most like to invest in?
There's so many in this space. I honestly, I think every single space. So I think forensics, I think in cancer, there will be big companies created in all of these spaces.
Google.
The companies that are focusing on the prevention of genetic disease through carrier testing, preconception carrier testing.
From what country other than the US will the next breakthrough in genomic sciences and big data come from?
UK.
Finland.
I'm excited about, the UK and the GEL project.
Other than healthcare, what's the most surprising application of big data in genomics?
I've touched on forensics a lot. I'm really excited about that.
CSI, right?
Yeah. Yes.
Plant biology.
Microbiome.
Present company accepted, who's doing the most interesting work in big data and genomics today?
Wow, there's a lot of very good. I'm going to say David Hausler at UC Santa Cruz.
Stephen Friend, Sage Bionetworks up in Seattle, is doing some amazing stuff.
Francis?
I think there's terrific work in big data and terrific work in genomics. I don't know yet that we found the great company that does the intersection.
And how would you advise someone who's not in this field to learn more and keep up?
Good luck. The good news is it's moving so fast that nobody's really an expert because they've been in it for a while. So the good news is we're kind of all in this together is the advice I'd give them. Read the News and Views and Science and Nature.
There are 3 great Coursera courses. One that Eric is a MOOC, a massively online course. One by Eric Lander, one from UCSF that I participated in, and a third from UCSF that Barbara Koenig participated in that's more involved in the ethical and legal aspects of it. Take those 3 and you'll know a lot.
Great. Thank you very much. Please thank our panelists.